Hi, dear participants. I'm Arzu Altunkaya Dinçay, Associate Professor of Food Engineering. Now I will talk about health benefits and risks of polyphenols. The outline of my speech consists of structural differences of polyphenols, dietary sources of each class, the factors influencing the bioavailability of the polyphenols, and some general allied health benefits and risks supported by current research. When we say phenolic compounds, they are plant-derived functional foods gaining considerable attention due to their safety and therapeutic potentials. Research on plant-based functional foods presents several challenges ranging from hypercholesterolemia to cancer prevention. These molecules consist of over 500 plant metabolites known as polyphenols that come from numerous food sources including various fruits, vegetables, grains, and beverages such as coffee, tea, and wine. In this context, more human studies are needed to provide clear evidence of their health protective effects and to better evaluate risks possibly resulting from polyphenol consumption. They are chemically diverse secondary metabolites which exert protective effects against various environmental stress in plants and generally involved in defense against ultraviolet radiation or aggregation by pathogens. And also in foods, the polyphenols may contribute to the bitterness, astringency, color, flavor, odor, and oxidative stability. And lastly, owing to an essential part of the human diet, long-term consumption offered some protection against development of cancer, cardiovascular diseases, diabetes, osteoporosis, and neurodegenerative diseases. When we have a look at the structure of phenolic compounds, they are the largest group of phytochemicals and their chemical structure poses an aromatic ring with one or more hydroxyl functional groups. These are classified into two sections, flavonoids and non-flavonoids. When we say flavonoids, they share a common structure of 15-carbon skeleton composed of two aromatic rings connected through a heterogeneous pyrrole ring. This carbon bridge structure can be denoted as C6, C3, and C6. You can have a look at the basic structure of flavonoids. When we say non-flavonoids, then they comprise simple polyphenols, phenolic acids, coumarins, xanthones, chalcones, cetylbenes, lignins, and lignans. In this group, some phenolic acids are found in free form in vegetables and fruits, but in whole, bran, and seed contains phenolic acids that in bound form. Bounded phenolic acids are released by acid-alkali and enzyme hydrolysis from brown seed. Another section I will talk about occurrence, content, and distribution of phenolic compounds. Distribution of phenolics in plant tissue, cellular and subcellular levels is not uniform. Insoluble phenolics are found in cell walls, while soluble phenolics are present within the plant cell vacuoles. Certain polyphenols like quercetin are found in all plant products, such as fruits, vegetables, cereals, fruit juices, tea, wine, infusions, etc. Whereas flavonols and isoflavonols are specific to particular foods. In most cases, foods contain complex mixture of polyphenols, and also the outer layer of plants contain higher levels of phenolics than those located in their inner part. When we have a look at the table, you can see that the more common examples of polyphenol food sources. For example, flavonols are rich in onions and tea, and isoflavonols are rich in soybeans and soy milk. Anthocyanins are in strawberries, cherries, and red wines. Hydroxybenzoic acids are rich in tea and flaxseeds, and hydroxycinnamic acids are rich in coffee, wheat, flaxseed, sesame oil, and red wine. You can see the average amount of polyphenols in these foods. And also some factors affect the polyphenol content of plants. These are degree of ripeness at the time of harvest, environmental factors, as well as adaptive factors like soil type, sun exposure, and rainfall. The another factor is processing, and the last one is storage. When I talk about the bioavailability of phenolic compounds, when we say bioavailability, it is a key step regarding functional foods and health claims related to food components, followed by knowledge of the circulating metabolites, which leads to understanding of the mechanism of action in relation to the benefit. In order to exert a health benefit, the compound of interest, whether hydrophilic or lipophilic, needs to withstand food processing, be released from the food matrix, post-digested, and be bioaccessible in the gastrointestinal tract, undergo metabolism, and reach the target tissue of action. The most abundant polyphenols in our diet are not necessarily those that have the best bioavailability profile. It means that it is not only important to know how much of a nutrient is present in the specific food or dietary supplement, but it is even more important to know how much of it is bioavailable. And also to establish conclusive evidence for the effectiveness of polyphenols in disease prevention and human health improvement, it is essential to determine the distribution of these compounds in our diet, estimating their content in each food, and also to identify which of the hundreds of existing polyphenols are likely to provide the greatest effects in the context of preventive nutrition. Lastly, it is necessary to know the bioavailability of polyphenols and their metabolites to evaluate their biological activity in target tissue. When we have a look at the table, you can see that the main factors affecting the bioavailability of dietary polyphenols in humans. The first one is food processing related factors, such as thermal treatments, homogenization, lyophilization, cooking, and methods of culinary preparation and storage. The second one is food-related factors, such as food matrix, presence of positive or negative effects of absorption. The third one is interaction with other compounds, such as bond with proteins or with polyphenols with similar mechanism of absorption. The fourth one is polyphenol-related factors. These are chemical structure, concentration in food, and amount introduced. The last one is host-related factors. These are intestinal and systemic factors. When we say intestinal factors, these are enzyme activity, intestinal transit time, and colonic microflora. When we say systemic factors, these are gender, age, disorder, or pathologies, genetics, and physiological conditions. The assessment of bioaccessibility and bioavailability of health-associated compounds is important for the understanding of the relationship between food and nutrition. Because of the synergistic effects of the mixture of polyphenols contained in each food matrix tested, definitive conclusions on the bioavailability and bioactivity of a single phenolic compound are difficult to obtain. Polyphenols and health benefits. Recent polyphenol research is primarily composed of epidemiologic cohort and case control studies that focus on disease endpoints in addition to mouse model and human feeding experiments that explore mechanistic interactions. Evidence generated by animal and human studies show that the antioxidant and anti-inflammatory properties of polyphenols may potentially prevent or serve as treatment against many non-communicable diseases. When we have a look at the health beneficial effects of dietary plant polyphenols, diabetes, we can say that diabetes, cancer, hypertension, asthma, infections, aging, cardiovascular diseases, and so on. The majority of evidence comes from in vitro pathway, and it is unclear if this mechanism holds true in humans. It means that it is important to do these experiments in humans. Risk associated to polyphenols consumption. Adverse outcomes have been documented from polyphenolic botanical extracts in beverages, especially for individuals with degenerative diseases, high blood pressure, thyroid diseases, epilepsy, or heart disease. Due to pre-absorptive interactions during digestion, dietary polyphenols have also been shown to reduce the transport of thiamine and folic acid, and to alter the activity of drugs through interactions that affect drugs transporter or enzymes involved in reactions, resulting in both inhibition and increasing the bioavailability depending on the case. And also, it's often said that polyphenol consumed in high amounts could have pro-oxidant effects. Such pro-oxidant effects have never been demonstrated in vivo. A pro-carcinogenic effect of quercetin in rat models of nitrosamethylurea-induced pancreatic cancer or azoxymetal-induced colon cancer have been reported. Moreover, adverse effects of polyphenol consumption on cardiovascular disease have recently been suggested. High polyphenol intake could increase the risk of cardiovascular diseases through an effect on homocysteine, an independent risk factor of cardiovascular disease. And as a result, more clinical trials and epidemiological studies are needed to better assess the balance between benefits and risks. In conclusion, consuming polyphenol-rich foods has been associated with a range of health benefits, including optimizing cardiometabolic health and to a lesser extent positively impacting brain functioning in humans. Much progress has also been made on the evaluation of their bioavailability. The significance for human health of all in vitro data obtained on cultured cells or isolated enzymes and receptors will have to be properly reevaluated in the light of this new knowledge on bioavailability. However, the actual mechanisms for these effects have not been fully established. We need to identify proper markers of disease risk and demonstrate that they are influenced by the consumption of polyphenols. And thank you.